In this video, we're going to talk about insect succession. So also a little bit of taphonomy and how stages of decomposition and forensic context can contribute to who is colonizing the remains and in which order. So succession being the time in which an organism comes to the remains. Majority of this video is going to be talking about who is there and when. And so who is also defined by what they do at the remains. So what they do is defined as functional groups because we can group them by what they're actually doing. So first I want to talk about necrophages. These are going to be the most common ones that we see. These are insects that are evolved to feed on decomposing tissues. So think of things like flies that are there to consume the tissue as maggots. Also, there's necrophagus beetles as well. You have things that are omnivores, so they're feeding on both the carcass itself, but other insects that are there. So there's a lot of beetle species that will do this particular type of activity. There's also things like ants and wasps that will also feed on both the tissues and other insects. There's going to be predators that are present. So these are organisms that are only feeding on other insects. So there are certain species of insects that are highly attuned to find carcasses. So they're using the smell of the carcass to go to the carcass, but they're only there to feed on other insects. So they're the ones that are going to reduce the numbers of insects that we're looking for to help determine our time of colonization. And then we're going to see something called advantageous species. So these have nothing really to do with the carcass itself. They're more there as a coincidence, but they're going to use the carcass as an extension of their environment. So they could potentially feed on the remains, or they can use it as shelter. They're not really attracted to the carcass. They're just kind of in the general vicinity. So again, these are going to be our major groups. I do want to specify that we're talking about terrestrial species. We'll talk about aquatic forensic entomology later in the course, but just for the purposes of this video, we're going to stick with terrestrial settings. Now again, our necrophagous feeders are going to be individuals consuming tissues directly. So our two major groups are going to be our forensically relevant groups, so both our Diptera. So this is going to be things like flesh flies and blowflies, in some instances houseflies. There's a lot of these big charismatic flies that are going to be utilizing the carcass. There's also a lot of really small flies that we'll find that can be forensically useful if our larger flies aren't present. There's also different types of beetles, more specifically the dermestid beetle, which is also called the hide beetle, because they're going to be the ones later in decomposition that are consuming the skin and the hair. Our common omnivores are going to be things like beetles. So the very top here is called an American carrion beetle, and also wasps. So they're going to come, they're going to be attracted to the tissues. They're going to feed on the carcass, but they're also going to feed on specifically maggots and other flies at the remains as well. There's also some flies that will actually consume other fly species. So this is a picture of Chrysomyia rufifasces. This is a blowfly species that as a larvae will feed on the carcass until about second or third instar, in which they shift into becoming predators. So they will exclusively, or for the most part, feed on other maggots once they hit this stage of this stage of larval instar. And once that other species is decimated, they'll finish off those tissues and move off the body. This becomes really important because in the instances where Chrysomyia rufifasces colonizes remains, they will be just about the only species found because they're going to consume the other individuals that are present. So this can influence our timelines because if Chrysomyia rufifasces came second, then we are actually missing the original colonizer. So our time of colonization may be slightly off in these particular instances. Now, Chrysomyia rufifasces is not the only species that does this. There are other relatives of the genus Chrysomyia that do this as well, and they're not found everywhere in the world. Usually they're found in more tropical areas, really warm, hot environments. So only in some instances, in certain geographic locations, is this going to be relevant. When we're talking about the insects that are only there to consume other insects, there are a lot of beetle species that will do this. Specifically, a group called the Staphylinidae. They're also called rove beetles. There are specific species that are specialized to consume flies, so they will be present. But there's also different parasitoid wasps that will find and colonize insect species, specifically those of the Diptera, so our flies. And so there are parasitoid wasps that their entire life cycle is dependent on finding a host, colonizing that host, ultimately killing them, and emerging in their place. So this is a picture on the right of Nasonia vitripinis. This is going to be a specialist colonizer of things like flesh flies and blowflies. So they will actually go to the carcass because, again, of the smell of the carcass. They will find a maggot that is about to leave the body to go pupate. They will follow along with that maggot until it turns into a pupae. They will lay eggs inside the pupae itself, and the offspring will feed on the fly as it's developing in the pupal casing. And in its place, the wasp offspring will emerge as adults, and the cycle will go over again. And then, of course, there's going to be advantageous species that are just around that will end up using the carcass as an extension of its environment. So this list is not all inclusive, but these are some of the more common ones that we end up finding. So things like scorpion flies and butterflies, and other things like harvestman spiders, or things that you might call daddy longlegs. They will end up going and even feeding on the material of or tissues and fluids that are on the remains, or maybe even using the carcass as a place to have a colony, for instance, or lay their offspring as a shelter. There are 5 recognized stages of decomposition, and this was actually not identified by an anthropologist, but an entomologist. And so he actually recognized these stages based off of the insect activity that was at them. And so it's not just the insect activity that is described, but it gives us really helpful insight as to what to expect when decomposition is being undergone. And so the main thing that you should know from this is that decomposition is not linear. You can't go from first stage to fifth stage to third stage. You can only go in logical progression forward. So fresh stage of decomposition is usually in those minutes to hours after the individual had died. This is when all of your cells undergo something called autolysis, and so that's programmed cell death. And also all of the bacteria in your gut is going to be starting to consume the tissues within you. So again, insects are attracted through chemical compounds to remains. Then the individuals that are producing those compounds are microbes, right? So those microbes are starting to feed, and as they feed, they're going to create metabolic byproduct that can be in the form of chemical compounds and gases. And so what ends up happening is the body starts to fill up with gaseous material. And once this happens, you've entered what's considered the bloat stage. Now, the bloat stage is usually when you're going to start seeing actual maggots. So the fresh stage, you will get eggs, you might have first instars, but the bloat stage is when those maggot masses are going to start forming and where they're noticeable. Once the body is completely stretched out to the point where the skin's elasticity ruptures, it now leaves the bloat stage. And what ends up happening is all the tissues within the individual ends up seeping into the surrounding environment. And so that actually creates this black material around the body. that's called the cadaver decomposition island, or the CDI. And so this initiates what's considered the active stage of decomposition, which is categorized almost exclusively by high insect activity. So large maggot masses, usually really large larvae, these are going to be mostly where the cases I get, the individual is in, usually in active or advanced decay. Usually the fresh stages, that's going to go to the medical examiner. They're going to be able to make an estimate because it's within usually that 72-hour time frame. Sometimes that's true in bloat stage, depending on the environmental conditions. And so active, you're going to have a lot of insect activity, mostly maggots, right? But you could have other consumers. In the advanced stage of decomposition, that's when you're going to start shifting away from the flies and go more towards those beetles, those late stage decomposing beetles. And they're actually going to be there because the skin ends up being a little bit more leathery. A lot of times it's just skin and hair that's left behind, and tendons. So they're actually coming for that resource. And then dry decay is actually when it's primarily skeletonized material. And so this picture on the side here is the actual study that Jerry Payne used to describe these stages of decomposition. What is really challenging is you will have individuals in like other fields use something called total body scoring to estimate time of death and things. The stages of decomposition and their actual times between each stage is highly variable, and there's a lot of things that go into it. So it is not a super reliable way to identify the time since death for an individual. So that's why we rely on insects. Now, our understanding of who shows up at the carcass and when they show up actually started with a scientist named Jean-Pierre Menin, and he ended up describing the groups that ended up coming and when they showed up during decomposition as squads, which later on ended up becoming seers. So throughout this video, I'm going to be calling them seers. And so what he had determined was that the succession, or these seers, and when they progress, ended up changing with the body treatment, but also based off of the season. So the succession that happens in the fall isn't necessarily going to be the same that happens in the spring, vice versa. And what I really mean by treatment, especially in earlier studies, I'm referring to either the individual is buried or they're placed on top of the ground. So early studies have showed that if the individual was placed above ground, that there's 8 distinct seers that have specific insects associated with them. And then in burial scenarios, we have 2 different seers with specific insects. Now, early on in forensic ento, we actually used insect succession to help provide a postmortem interval. And it's really important that we don't really do that anymore. The reason why insect succession is important now is because it helps us get a better understanding of why a specific insect is on the body at the time of collection. So while we have these seers, and we're going to talk about what changes the seers and the conditions, it's really important to note that the time of colonization that we estimate is not based off of this succession. What I also want to make really clear is that succession is a continuum, and just as the stages of decomposition, it is linear. And so that means we can't really go backwards in time. There's certain species that are going to show up in later stages of decomposition because of the tissues that are available. We're not going to be able to get individuals that prefer a lot of the fresh tissues in these later stages of decomposition unless there is a specific treatment of the body that rehydrates the body in some way, or some type of external circumstance happens that allows for recolonization to happen again from these early colonizers. And there has been a lot of research looking at what insects show up at different stages of decomposition. So all of our charismatic fly species that we're going to use for our forensically important timelines are well established to show up early on in decomposition and hang out during those more active times where you're going to be collecting a lot of these fly larvae. Then we have a lot of these other insects that will show up later on in decomposition. So, depending on when you have discovered the remains, you're going to see either early colonizers or late colonizers just based off of the conditions of the body. One of the main reasons why we don't use insect succession for our forensically important timelines anymore is that there is a high degree of variability of these seers because of abiotic conditions that can influence what insects are around at the time of the individual becoming deceased. So, I wanted to go over just some of these things. So, one of the things that are really important is called spatial variability. So, depending on the location in which the remains are, we can have different insects that are present. So, there are some species, especially different fly species, that can be more urban than rural. So, if you have remains placed in the middle of the city, we might have different insects that are present than in rural settings. This is especially true as decomposition progresses. If you have an individual on concrete, it's going to be very limited in what insects have access to those remains versus in a wooded habitat. We also have to consider things like forested landscapes and open fields because just as in urban and rural settings, the insects that are going to be present are going to be different just because of the environment that you're encased in. So, in forest settings, we're going to see a lot more diversity. Also, what's really important is the amount of sunlight that the remains encounters is going to change the temperature around the body, and it can actually influence what species is present as well. We also have this idea that we can have aquatic scenarios or burial scenarios. Now, I have a YouTube link here showing the progression of decomposition in submerged scenarios that you can watch at your own leisure. But what's really important is that decomposition process is going to be very different in these different scenarios. And because of access to the remains, we're going to have different insects that are present. Especially in aquatic scenarios, we're going to have a lot of different types of arthropods like crabs and other crustaceans that are going to be present in place of terrestrial insects. We also have different geographic locations. So, depending on what continent you're on, you're going to have different species just because of who's living in these different locations. So, it's really important that when you are looking at a particular case, even in different states. So, if you're in the United States and you have a case in Texas, it's going to look very different than cases that exist in Massachusetts. So, it's really important that geographic location is also taken into consideration when we're looking at who is going to show up at a body and when. So, especially when we're talking about seasonal variability, if the remains are really dry, you're going to see a lot of those later colonizers that prefer those dry conditions showing up earlier on than in situations where there's high humidity that might push the colonization of these individuals that prefer drier conditions later on in those stages of decomp. We also have different forensic conditions. So, if remains are placed indoors, there's going to be barriers to entry for insects. So, it'll limit the amount of insects that actually interact with the remains in comparison to outdoor scenarios. There is also temporal variability that we need to consider. And so when remains are placed out, depending on when during the day they're placed, can influence when insects have access to it. So, insect activity for forensically relevant insects, such as our flies, are likely not going to happen at night. But in some situations where there's some external light, that can happen. So, it's not a perfect science. But generally speaking, we don't expect colonization to take place at night, but we would during the day, especially during peak hours of the day where the ambient temperature is at its warmest. Another thing that we need to consider is seasonality. Depending on what time of year remains are placed out in the environment for insects to have access to, we're going to expect to see different insects. So there are certain insects that do really well in cooler temperatures, and there are some insects that do really well in extreme heat as well. So depending on that time of year, we're going to see different species that we would expect to be colonizing the remains. This is even true at really refined levels of taxonomy. So even things like blowflies, there are many different species of blowflies, and some of them really prefer warm temperatures and some of them prefer cool temperatures. So depending on the time of the year and the geographic location, we're going to expect different species of forensically relevant flies to be on the remains. So it's really helpful to know what temperature or what season we are in to help give an estimate of what species we would expect to see. Conditions that influence the insects that are present doesn't have to be abiotic. It can also be directly related to the remains themselves. So, individuals that are on the smaller size are going to have very different insect communities than those that are larger individuals. This also is in consideration with things like fat content and muscular content and things of that nature. So it's not just, the individual is lighter versus heavier. There's other things to consider when we're talking about the individual characteristics themselves. And of course, in forensic contexts, individuals are going to try and conceal remains or treat them so that way they can cover up the crime. And when individuals do this, it can influence the insects that colonize the remains. So, depending on how badly burned an individual is, it can limit the amount of oviposition sites and change what insects are willing to colonize the remains. And in situations where remains are attempted to be concealed, such as in wrapped or buried or placed in different containers, it's going to create these additional barriers to entry that can prevent certain insects from having access to the remains or delay that colonization, interrupting how those sears will take place later on. Ultimately, when we're looking at forensic context, there's going to be a mixture of different factors going on. So you can have an individual who's wrapped in a house in an urban setting, and that's going to have very specific insects on them. So really, what we're doing in forensic casework with entomology is putting on an ecologist's hat because we're identifying the ecological niche of that organism. But okay, that's all I have for you today.